thanks everyone for joining the TechTake presentation hosted by NCI this morning. Today we're grateful to have two speakers, Matthew from Associate Director from NCI and Johan from Australian Bioplatforms. Without further ado, I will hand it to Matthew.
Thanks, Fred. So this morning I'd like to talk about some of the work that we've done here at the NCI to improve the experience of users who have sort of bioinformatics workflows here at the NCI. I'm going to break this down into a couple of different parts. I'm guessing that most of the users here, or most of the people on this call, are actually already users of the NCI facility, but I will just give a high-level overview of the computing resources here. Then I want to talk about some of the work that's been done to improve access here, then go through various things that have been done to improve data analysis environments, then software installation and workflows. And what I want to emphasize here is that this has been a large-scale community effort involving a number of users and user groups. Particularly like to mention the Australian Biocommons, who we've worked with quite closely on a lot of this work. But we've also done work with users and, as I said, users and user groups at places like Sydney Informatics Hub at the University of Sydney, John Carton School of Medical Research here at ANU, and elsewhere across Australia. So NCI is the National Computational Infrastructure. We are a Tier 1 high-performance computing facility that's based in Canberra on the ANU campus. The core resource that we have here is a supercomputer called GADI, which has around 180,000 cores across a mixture of CPU and GPU nodes. And there's also one thing I'll point out to is there's a number of high memory nodes which may be useful for certain sorts of analysis. And we have, in addition to that, there's cloud resources in the form of Near the cloud here that was mentioned in the previous TechTake. And backing all of this is a very large, a number of large Lustre file systems which provide over 100 petabytes of storage. And we also have more storage coming online as well. So in terms of access, access for things like bioinformatics projects can be a bit of a pain, particularly in cases where the data generation step may have sort of uncertain timings. There's a number of merit-based access schemes that are available for users to access the NCI facilities. I've got two of them up here. The first is NCMAS over here on the right-hand side, which is sort of designed for larger projects. It has quite a lengthy application process. Projects are 1 year in duration, and there's an annual application cycle for that is currently open and finishes in about 3 weeks' time. NCMAS, as I said, is annual, so it takes an annual application cycle, which may not be suitable in every case. Recently, over 2022, the adapter scheme has started up, which is designed for flexible access. It's really designed for shorter duration projects of up to 250,000 SUs per quarter. SUs are the unit of compute that we use here, and it takes quarterly proposals. So the cycle for proposals, which will begin in the last quarter of this year, is just closed, and there will be another one opening up later, which will be for the start of 2023. So, as I said, we've done a lot of work across various aspects of the sort of research life cycle. One place where we've put some effort in recently is in interactive data analysis. So you may have generated a large amount of data, and rather than pulling it back to your host institution, or before pulling it back to your host institution, you can do some data analysis just to check over that data. And we've got two environments that are useful for this. They're both based on the same technology. The first one and the preferred one is the ARE, which stands for the Australian Research Environment. So if you're a user of the NCI facility, you can go to this URL here and get access to it. It's a web-based system that gives access to data analysis environments, which are backed by Gaudi nodes and storage. So if you'd like to use GPUs for some interactive data analysis, perhaps visualization, it can be done there. There are two main applications that are installed in ARE. The first is a virtual desktop environment. So you can basically set one of these up for a set period of time and do things like interactive visualization of results. For bioinformatics in particular, IGV is installed in this environment, so you can use that for looking at alignments and other aspects of research. There's also a Jupyter Notebook environment that's been installed, which allows sort of access to notebooks, so you can mix calculations and the outputs of those calculations, which might be graphics, as well as text editor and backing with a terminal as well. To sort of help users make use of these environments, we've created a number of software projects that contain all of the or many of the tools that you might commonly want to use. There's two in particular that I'll highlight. The first one is a generalized data analysis project, which goes under the short form DK92. This includes Julia, Python, and R environments and a number of packages. And there's also a more bioinformatics focus project called HR32, and I'll come to that in a slide later. There's also an older form of interactive environment called OOD. So ARE is our current preferred environment, but OOD is still around, and this has the same core applications as the ARE, but it's sitting on cloud hardware, which means it has a couple of downsides. One is limited access to file systems, but one of the upsides, and one of the reasons it's still there, is that longer wall times are available, and we've also created a Cromwell app, so you can set up the Cromwell workflow manager and use that for longer wall times, up to a week in this case. So I said I'd come back to the bioinformatics analysis software project, HR32. This has an R environment and a Python environment, and I'll get to those in a moment. So this can be used from both the command line and from Jupyter Lab environments. It contains common command line tools that might be used in bioinformatics, such as GATK and SAM tools, and a set of commonly used R and Python packages that we've found by consultation, or we've identified by consultation with both user groups and by looking at other environments such as the Terra environment. So this is open to all users. You can just go through, anybody can join it and use the resources that are available in it. It just provides software, but doesn't provide any other resources available. And currently this is due for an update. So if there are any current users who might have some feedback on how well it's worked for them, I'd really appreciate it if you can drop me a note, and we'll incorporate it in the next update. So one of the problems with certain bioinformatics tools is that or pieces of software is that they might involve slightly more complicated steps to install centrally than can easily be accommodated here at the NCI. And so with the Australian Biocommons, we've created a tools repository, which has the project code IF89. And this uses the NCI framework for software installation. And it's really a place where packages that, as I said, are difficult to install centrally can be installed. At the moment, there are about 60 packages installed there. We're exploring how we might include common data sets and how workflows might be installed. And as with HR32, this is open to all users. In terms of improving the visibility, I'll point out one tool, which is Tool Finder. Johan, who's going to talk after me, has played a key role in setting up. So this is not actually an NCI tool, but it's an Australian Biocommons tool. But it's a directory of all of the relevant software that's installed at various sites across Australia. So any package that is of relevance to the bioinformatics community and is installed at NCI will be automatically shown within the tool finder. So it can be a good way of searching out what's available. And that includes both centrally installed applications in /apps directory, as well as tools that are installed in IF89. So I've gone through access, data analysis, as well as software installations. The last element of the list is just workflows, really. So we've done a bit of work to support 2 workflow managers. The first of these is Nextflow. Nextflow is a popular workflow manager that's both a workflow manager and a workflow language. And one of the nice things about it is that it has a very clean separation between the workflow and the resource specification. So what I mean by that is that, when you request resources as part of a workflow, you may ask for a particular package to be installed, or to use a particular package or module, or a particular amount of memory, or put things through a particular queue. And then Nextflow, those are very cleanly separated from the actual workflow definitions. And a real upside of that is that it makes the workflows more portable, so that they can be developed in one place and then ported to another place relatively easily. Here at the NCI, we've installed a slightly modified version in the /apps directory that has improved support for the PBS Pro queuing system. So it has a couple of extra directives to allow you to specify the project storage and GPUs very easily. And so here's an example of that. So here's the resource definition. You pull out, you specify the executor that you want to use, the PBS Pro, queue things you want to send it to, the project code that you want to charge resources against, as well as any storage that you want to use. And when Nextflow goes to submit a job to the queue, these will all be formatted correctly in the PBS Pro directive. If you don't want to use these, it's still possible to use the cluster options directive. All of this is documented at this URL here. Finally, I just want to talk about Cromwell. So Cromwell is a workflow manager for workflows which are written in the WDL, or workflow description language. We've put in improved support for PBS Pro with a particular config file that's available for users. And longer wall times are also supported through an OOD app, which will start Cromwell, and then you can interact with that Cromwell workflow manager through the command line using a tool like Cron Shell. And that's all documented at this URL here. So just to summarize, we've been working quite hard to improve the bioinformatics user experience by addressing a number of pain points around access, software installation and software visibility, data analysis, which is a key part of the research life cycle, and then also workflows as well. And with that, I'll close up, and I think we'll take questions at the end of the presentation after Johan's presentation.
Thanks, Matt. Okay, so my name's Johan, and I'm part of the community engagement team from the Australian Biocommons. I wanted to present on making workflows findable, and the things I wanted to cover: first, a brief intro to the Australian Biocommons, and then straight into workflows. So the why behind making them discoverable, workflow registries as a solution to that, and then finally a crash course in Workflow Hub. So starting with the Australian Biocommons, in essence, we engage broadly with communities. And so you can see some examples of those here: genome annotation, genome assembly, whole domains like proteomics and metabolomics. And we facilitate interactions with computational providers. The reason for this is that we want to build a more fit for purpose, flexible and cohesive data analysis ecosystem. And in essence, we're advocates for community requirements. To do this, we work with many partners. And so here's some of those partners up on the screen, and you can see NCI is one of our key partners. But this also extends to other computational facilities, institutions, as well as Elixir in Europe. Okay, so why would you want to make a workflow discoverable? Well, first of all, just in case I needed to point it out, workflows are important things. They allow you to construct a complex analysis and make it reproducible. It's something you can share with others. It is something that will reduce replication of effort and accelerate science. And if we look at some example numbers from the broader community, there are more than 300 workflow languages currently. And if you look at the number of clones for a really popular workflow on Nextflow Core, you've got more than 400,000 clones for that workflow. So they're clearly important artifacts. But they can also be complex. They can be time consuming to build, and maintenance can be a real issue. They can also be difficult to find, difficult to redeploy. And unfortunately, a lot of workflows end up duplicated or replicated. So people are building the same workflow over and over. Because of these reasons, because of the amount of time that gets poured into building them, because of how important they are to reproducibility, workflows really should be findable. They should be reusable, and they should also be citable. And what I'd like to propose is that workflow registries are a way to address just that. So there are multiple places that you could find workflows at the moment. So if you're aware of domain-specific platforms or community repositories, you can go straight to one of those to search for workflows. And some examples of those are Galaxy and Nextflow Core. You can go straight to GitHub. You can, of course, look in the literature, and you can also search Google. But what you'll notice is that this is quite a distributed set of options you have for finding workflows. The reason that a registry is ideal is that it's a central place. It's something that's searchable. It's something that is ideally integrated. You have a standardized set of metadata. You have citable artifacts, so you can generate things like object identifiers. But you can also be interoperable with other platforms. So think of the concept of launching a workflow directly from a registry. At the moment, there are two key options for workflow registries. The first is DocStore, and the second is Workflow Hub. Now, if we look at some numbers in terms of utilization of these registries, you can see that in DocStore, you've got more than 1,600 workflows. If you look at an example like Galaxy Europe, which is a big instance of the Galaxy platform, there are 732 published workflows and more than 250 in Workflow Hub. So this is quite a small fraction of the workflows that would be available across the community. So out of those two big registries, DocStore and Workflow Hub, I wanted to spend a bit more time talking about Workflow Hub because that's one of the platforms that we collaborate quite closely with. And I wanted to start with the high-level concept behind the hub. And it's basically that you register, you select a space, which is the biggest organizational unit on the platform. You either create or join a team, and then you register your workflow. So going through step by step, the registration is the first step. You'll notice that you can actually log in using existing credentials. So this isn't something that you need to generate a new set of credentials for. You'll choose a space. So I've given two examples here of spaces. One is the Australian Biocommons space. All Australian researchers, if they'd like to have a space, if they'd like to have a team on Workflow Hub and they're not sure what space put it in. They can actually request one in the Australian Biocommons space. And you've also got independent teams. So this is if you just want a team, which is just maybe your research group or your project, you can just create a team within that space. So once you know what space you want to be in, you can join a team or create one. So I've just put up an example here of the Galaxy Australia team. And then finally, you add your items. And this isn't limited to just workflows. You can also add things like publications. You can add any document effectively, standard operating procedures. You can even create collections of these items. But the key thing I wanted to talk about today is the workflow itself. So the workflow registration process has an inbuilt wizard on the platform. So it's really straightforward to add workflows. And it's a 3-step process. The first one is to provide a workflow file. There are multiple ways of doing this, but the most straightforward and the recommended one is actually just to provide the URL to the Git repository. So there's an integration between Workflow Hub and repositories like GitHub, so that you can point to that URL and it will import a lot of material from the repository into the registry. The second step is to add some additional metadata, and these are things like title, the description, the license, any tags that you want to add, and then indicate what team it's associated with. And then finally, your review. So this is a Workflow Hub registry entry for a workflow called germline short biovalidation. And so you can see here a description on the left. On the right, you basically have all the associated metadata, starting with creators and submitters, the citation, license, some information about activities. So this includes things like views and downloads, but then the tags that you've assigned. So you can see down here, there are EDOM ontology topics and operations that have been added for this workflow. There are buttons at the top, so you can go directly to the GitHub repository as well. All right, so instead of showing you more slides, what I thought I'd do is for the search and update, I would actually do a quick demo of the platform. So we'll jump over to Workflow Hub and we'll go to the main page. So this is the main page that you would see when you visit the platform. So if you want to search for workflows, basically you can click on the big Discover button and that will take you straight to the workflows that are available. In terms of searching for these workflows, it's quite straightforward. You can use any of the filters down the side. So things like workflow types, tags, the submitters, teams, spaces, all the way down to things like ontology, topics and operations to filter. So we might be interested in sequence assembly. And then we can see the workflows that are available for sequence assembly. You can also do a direct search as well. And that will allow you to find the workflows that you might be after. And then you can make use of the filters again. That's searching for a workflow. So that's quite straightforward. In terms of updating workflows, if you need to maintain your workflow at the moment, the options are to go to the workflow that you actually own or are responsible for. And under actions here, this is basically where you can do all the edits and upkeep for the registry entry. The Workflow Hub team at the moment are working on an app that will strengthen the integration between GitHub and the registry, so that when you push a release on GitHub, you won't actually have to come back to the registry. All right, so just to summarize what I've gone through, the platform allows you to search for and discover workflows. It is accessible via API. It doesn't actually replace your development repository, so that's not its purpose. And that's why you have the integration with Git. You have the ability to mint digital object identifiers, which is quite powerful if you want to include a citation in a publication direct to your workflow entry. You have the ability to put not only EDOM ontology tags in, but also custom tags to make it easier to find your workflow. It provides a set of standardized metadata, the Git integration, like I've already mentioned. You can actually add multiple artifact types. And because of the way it's designed, you can link all of these things together. So you can associate publications or documents or other things to workflows and vice versa. And it has an open development principle. So there's a team called the Workflow Hub Club that meets every fortnight. And that's basically a place to talk about further development of the platform and building the community. So thanks very much. I'm happy to take any questions, including after the presentations. You can email me at the address I've provided on this slide. Thank you.
Thank you, Johan. Thanks, Matthew, for this two informative and useful talks. So any questions, please feel free to ask. I got one. My background outside is biocom, so workflow is quite a foreign language to me. So I'm just trying to understand better about this idea. Okay. So when you mentioned workflow, I suppose this is because of this sort of intrinsic feature in this area where you got like a very large data set, complex data set that have so many tools. And to interpret and parse this raw data set, I guess you have a sequence of steps using tools and it generates meaningful results. Is it this sort of what workflow is aiming for?
So the definition of workflow can actually be quite broad. And so it's everything from a set of tools that have been strung together with a workflow language like Nextflow through to just a series of bash scripts that represent the steps that you've taken. So for a registry like Workflow Hub, the principle is that it's workflow type agnostic. So it doesn't really matter what you've written your workflow in. The principle is that if it's something that you can share that would be meaningful for other people, you probably should share it.
Right. But if I change, can I understand workflows as sort of like functions of composited functions that mapping from like your input to output now? That's way too simplified.
Yeah, so you would put together a workflow where you wanted to repeat an analysis. You know, if you find yourself doing the same thing over and over with the same sequence of tools, that's probably a time when you want to build a workflow. So there's still a significant part of the community that does things one tool, then they go to the next tool, then they go to the next tool, right? So that's still a way that the community makes use of bioinformatics tools. But when you have a use case that needs you to be able to reproduce an analysis or be able to share a way of working with other people, a workflow is a good solution.
Right. So in other words, of automation of this process is going through the tools sequentially. Is that common that people are working on simplified workflow sort of tree structure by, if there's a 10 different tool, if you're changing, so if there's a chain of tools, if you change some blocks in this chain, and would they still produce the similar results? Like this, there's something people working on in this area. Like I'm saying, like replacing some tools in this workflow with a simplified tool, and it gets the similar result.
So if you change the tool, you obviously can't guarantee that you'll create the same result. If the tool is doing something similar but doing it in a better way, you might be creating a similar result, but it wouldn't be the same.
Right. Okay. So it's kind of unique. Okay. So if there's no further questions, anything else, Matthew and Johan, you want to add?
No, I haven't really got anything.
Okay. I think I'll probably just call it there. Again, thanks for your participation, especially thank Johan from Australian Bioplatforms and Matthew to give this one talk.